So here you can see the picture here. At this corner looks a kind of complex threading complex structure. Okay, so this looks like glycogen structure inside the body. How glycogen present in the form of branched structure. Okay, I'm going to talk about this, how they are going to form as a branched structure, and what is the advantage. What is the advantage when glycogen or polymer has branches in its structure? We are going to see here. Okay, so if you see glycogen, as you see that glycogen is a polymer of sugar, particularly glucose. Okay, particularly in animals and fungi and bacteria. Okay, and when you see the polymer of glucose in plants, particularly you can see starch and cellulose. Okay, so glycogen is a polymer of glucose. We can call as polymer of glucose, and it can be used as a kind of storage of glucose inside the cell for metabolic activities. When glucose levels are more, animal cell can synthesize glycogen for later uses. When glucose levels goes down, glycogen can supply its mono units from its structure. Okay, in animal, a constant supply of glucose is essential for tissues such as brain as well as RBC. Okay, so these brain tissue and RBC totally depend on the glucose only for their metabolic activities or for their regular activities. Okay, and liver is the main source of site of the glycogen storage. Okay, liver glycogen is enough. Okay, whatever the storage is enough for off day when we are in starving condition. Okay, so under fasting conditions, as we seen in previous class, okay, when glycogen levels goes down, when no glucose levels are present inside the cell, so cell will synthesize the glucose from the non-carbohydrate molecules via gluconeogenesis. We have seen that in previous class. So here the structure of glycogen looks like this. You can see structure at this corner, branched structure. So the chains has two kinds of bonds here. One is alpha 1,4 glycosidic bond. Another one is another branch, another structure we can see here, alpha 1,6 glycosidic bond. Okay, so this linear structure will have 1,4 glycosidic bond, alpha glycosidic. Wherever the branch is created, that and this glucose will have alpha 1,6, and alpha from this branch, wherever adding the glucose molecule, will have the 1,4 glycosidic bond, and wherever the structure which branch point is the glucose, that glucose will have next to glucose with the alpha 1,6 glycosidic bond. Okay, so that's why you can see branches instead of linear polymer. You can see glycogen has kind of branched structure which is having two types of glycosidic bonds. One is alpha 1,4 where it is linearly linking, and wherever the branch point is required, wherever the branch point is generated, that branch point will have 1,6 glycosidic bond. Okay, that is the branching points, and you can see that's the region glycogen has many branches here. Just the cartoon shows how branches are created in glycogen. Okay, so as I said, glycogen is present mainly in liver. Main storage point is liver, and some extent it is also present in muscle cells also, skeletal muscle cells also. Okay, so glucose can be rapidly delivered to the bloodstream when needed. Glycogen. Okay, so that's the reason. That is the reason you are seeing branch points glycogen structure. So whenever it is needed, if it is linear, it is only one glucose will be removed. When it is branch points, from all branches, one glucose molecule can be removed at a time. So rapidly you can get lots of free glucose molecules when glucose is required from the glycogen. So that it is called as glycogenolysis process, degradation of glucose, removal of glucose from glycogen, it is called as glycogenolysis. And when enough glucose are present, when high levels of high levels of carbohydrate means glucose is present inside the cell, so the free glucose will be synthesized as a polymer, converted as a polymer. That process we can call as glycogenesis. Okay. So in this glycogen metabolism, you can see one is synthesis and another one is degradation. When required, that time glycogen will be released as a glucose molecule, that it is called as glucogenolysis. When glucose molecules are rich in bloodstream or rich in system, these glucose molecules are taken and synthesized as a polymer, that it is called as glycogenesis. Okay, so now we are going to see glucogen synthesis, glycogen synthesis. Following enzymes reaction occur in glycogen synthesis. First one is glucose activation. When glucose levels are high, glucose need to glucose glycogen. Okay, for this process, glucose need to get activated. And once glucose is activated, initiation of glycogen synthesis. Once glycogen synthesis is started, chain elongation. So once chain elongation, linear alpha 1,4 structures are synthesized at a certain number, so immediately another point it has to synthesize so that more glucose molecule can add to this polymer. So some enzymes involved in the introduction of branch points. Okay, so once points added, so many glucose molecules can be synthesized, can be added to these branches. Okay, that is the advantage because of this having branch points in glycogen structure. So now, glycogen biosynthesis, glucose is transported into liver cells by a specific glucose transporter protein. We have seen in glycolysis how glucose molecules enter into the cell and how this glucose molecule trapped inside the cell. Okay, so some proteins are, those proteins are called as glucose transporting proteins, and these proteins transfer the glucose from outside to inside. So when glucose is not phosphorylated inside the cell, it again transport back to outside. So so some enzymes are there, those enzymes immediately phosphorylate them. That, for example, glucose kinase and hexokinase are the enzymes present in the cell. So they make glucose into a form so that glucose 6-phosphate or the phosphorylated glucose cannot transport outside. Okay, so most glucose in cell is in the form of glucose 6-phosphate because of these enzymes, glucose kinase as well as hexokinase. So now here glucokinase, the glucose 6-phosphate has to convert into glucose 1-phosphate to synthesize the glycogen. Okay, glucose 6-phosphate is not substrate for the glycogen synthesis. So glucose 1-phosphate is the substrate for the glycogen synthesis. So simple mutase enzyme can convert glucose 6-phosphate to glucose 1-phosphate. Okay, this is a reversible reaction. Whenever glycolysis, glucogenesis is there, glucose 6-phosphate will convert into glucose 1-phosphate. When glucogenolysis is there, then glucose 1,6-phosphate can convert back to glucose 6-phosphate. Just like glycolysis and gluconeogenesis pathway. Okay, so once glucose 1,phosphate is synthesized, it will it will become uridylation. UDP will add it to this glucose 1-phosphate and it become UDP glucose molecule. Okay, so here you can see UTP is the one that donate UDP structure by removing pyrophosphate molecules from this. Okay, so here phosphate molecule will be removed from 1-phosphate molecule from UTP and 1-phosphate molecule from glucose 1-phosphate. Okay, so it become UDP glucose in the presence of UDP glucose pyrophosphorylase. So you can see a removal of pyrophosphate molecule from its structure. Okay, so this phosphate molecule pyrophosphorylase will be added in organic pools by pyrophosphorylase by breaking squiggle bond between these two. So inorganic phosphate molecule enter into inorganic phosphate pool. Okay, so this is not important here. What is important? Conversion of UDP glucose molecule, which is important substrate molecule, activated glucose molecule for synthesis of glycogen. So this step is conversion of glucose 6-phosphate to UDP glucose. Phosphoryl transfer is there. UDP as equal to ADP energy rich molecule. So energy breakage is there, energy liberation is there, energy used to activate the glucose. Okay, 2 phosphates from UDP are lost as pyrophosphate. Okay, so pyrophosphate then broken down by phosphate into inorganic molecules, and those goes into inorganic phosphate, okay, inside the cell. So this is the committed step in the glycogen synthesis. So now once UDP glucose is synthesized, okay, it has to bind to one protein molecule. Okay, so that small protein inside the cell is called glycogen. This protein, in this protein, tyrosine hydroxyl group of this protein acts as a seed for this glycogen synthesis. So this UDP glucose molecule interact with this molecule, tyrosine hydroxyl group, and it will create a bond, glucose subunit attached to 4-hydroxy group of the first one, and then repeat it over and over, give rise to a long linear polysaccharide. So now this UDP will be cleaved here. UDP will be the UDP glucose will to hydroxyl group of the tyrosine molecule of this glycogen, and then next UDP glucose will come and interact with the hydroxyl group of the first glucose molecule. So that will create a linear polymer when UDP molecules come and interact with the growing. Okay, so will create a non-reducing glycoside like an alpha-1,4 glycoside configuration. So this will create a kind of a long linear polysaccharide. Okay, this creation will be done. Creation will be this long chain polysaccharide creation. Generation of these long chain polysaccharides will be done in the presence of an enzyme which is called as glycogen synthase. Okay. So here you can see glycogen synthase. Enzyme is glycogen synthesis. Enzyme is glycogen synthase. The glucose which is attached to hydroxyl glucose, a hydroxyl molecule of tyrosine in the glycogen, and glucose molecule come and interact UDP glucose and come interact with this, and glucose molecule will be attached to this by removal of UDP, and glucose molecule will add it to this glycogen. So you will get glucose N+1. Okay. So here you can see the growing structure. So this is the structure where initial UDP glucose molecule is attached to hydroxyl group of the glycogen, and UDP glucose molecule will come and interact with here 4th carbon molecule and 1st carbon molecule, and you can see alpha-1,4 linkage here. Okay, so UDP will be released from this structure. So glycogen synthesis is inhibited by phosphorylation and regulated by glucagon. We'll discuss later this in the regular. So next, once addition of glucose molecule at the 1,4 linkage. So once the addition reached 6 to 7 residues. Okay, so immediately branching chains get activated and they look for to create a branch point, and they involved in creation of branch points after 6th or 7th residues of 1,4 linkage. So they add this upcoming UDP glucose to has linkage. Okay, so one six linkage is added to that residue, and then you can see two ends will be present to add glucose molecule once UDP glucose is sufficient inside the system. Okay, so like this, once another 6 residues are added having 1,4 linkage, another branching enzyme create another linkage for 1,6 linkage, so that another 3 points will be there to add glucose molecule in the polymer. Okay, like that, many branching points will be created branching engines, so that will have many sites to add activated glucose inside the glycogen structure. Okay, so because of branching points, accelerate rate of the glucose release during the degradation when glucose levels are required inside the cell, so that enzymes will interact with these branching points, so that they will release the glucose molecules. So many glucose molecules can release at a time from the glycogen. Okay, this is the advantage having the branch structure inside the glycogen. So here you can see growing chain. This is the seed growing chain 1,4 linkage. So once it reaches 6 to 7 residues, branching engine will add branch points. So once branch engine add one more branch point, so they will have two sites to add glucose molecule here and here. So once they reach 6, residues, and again branch points can be added, branching engines can be added, branch points, so that like that new branch will be there to add more glucose molecule, activated glucose molecule inside the structure by branching engines. So here you can see glycogen synthesis, linear polysaccharides, glucose residues, again using UDP glucose as a substrate. So this is the glycosamine protein, tyrosine molecule added. Tyrosine molecule is the group will be attached to glucose molecule, and then the UDP glucose, which is activated UDP glucose, interact with the glucose molecule. Upon losing UDP, the glucose will attach to this molecule, and linear polymer 1,4 linkage will be generated. So once after certain number of glucose residues are synthesized, branching enzymes can be make this another branch point, 1,6 branch points, so that it will have another point to add new glucose molecules to that structure. Okay. Okay, any doubts so far?
No, sir.
Okay, so this is the way the glucose will be added to polymer structure of glycogen, and number of polymers will be number of branch points will be present inside the glycogen. So until glucose levels goes down, from the system, extra glucose will be used to synthesize glycogen. Okay, so now once glycogen is synthesized, you can see glycogen main site is liver and skeletal muscle. So glycogen is present in these tissues, and once once the glucose levels goes down inside the system, so immediate source for the glucose molecule is glycogen inside the cell. So immediately glycogen degradation get activated. Okay, so one sec, my research student is calling. Okay, now glycogen synthesis we are going to see. So few enzymes are involved in the glycogen degradation, which is called as glycogenolysis. Okay, so the first enzyme which remove the glucose molecule from the glycogen is glycogen pyrophosphorylase. Okay, so this enzyme interact with the non-reducing and non-reducing sugars present in the mostly the sugars present, end sugars will have non-reducing ends only. Okay, so they interact with this end sugar and it will remove the sugar molecule from the glycogen structure as glucose 1 phosphate. Okay, so glucose 1 phosphate will be removed and pyrophosphorylase, glycogen pyrophosphorylase use pyrophosphorylase and it will cleave it will inorganic phosphate, it will create glucose 1 phosphate. Okay, enzyme is glycogen phosphorylase. Okay, glycogen phosphorylase, not pyrophosphorylase, glycogen phosphorylase. Okay, so now glycogen is a polymer that we have seen and which is having alpha 1,4 and alpha 1,6 glycosidic branches, and it occurs intracellular as intracellular granules, approximately 100 to 400 angstrom diameter, and each granule will have at least 1,20,000 glucose units in it. Okay, so 1,20,000 glucose units present in single small glycogen granule. Okay, so like that, many granules are present inside the liver cell, many granules are present inside the muscle cell. Okay, these granules are prominent in muscle and liver, and then once it is required they will start degradation. Once it is required they will start synthesizing the glycogen molecule. Okay, glucose units are mobilized by their sequential removal from the non-reducing ends of the glucose. The ends lack C1 hydroxyl group, which is called as non-reducing end. That is very primary thing. Whereas glucose has only one reducing end, there is a non-reducing end on every branch. Okay, glycogen is highly branched structure, therefore permits rapid glucose mobilization through the simultaneous release of glucose units from every branch. As I said, few enzymes are involved in this glycogenolysis. Few means 3 enzymes. One is called as glycogen phosphorylase, simple phosphorylase reaction, catalyzed glycogen phosphorylases using inorganic phosphate from the inorganic phosphate pool. So it removes the glucose molecule from the glycogen as glucose 1,6,1 phosphate. Okay, so this pyrophosphorylase is a dimer enzyme which is having for 842 97 kilodalton subunit, and it present in two. It get activated. It present in 2 forms. One is activation and inactivated form. This activation can be done by covalent modification as well as allosteric interaction. Okay, so phosphorylase A form is phosphorylate group esterified at serine 14 amino, and D phospho group is called as phosphorylase B. Phosphorylase allosteric inhibitors are ATP, glucose 6 phosphate, and glucose molecule. These are inhibitors for this enzyme. Okay, so active form, which is called as phosphorylated form, which is called as phosphorylase A. This active form, conversion of active form phosphorylase A, is inhibited by allosterically ATP molecule, glucose 6 phosphate, and glucose molecule. Okay, because when glucose molecule is high, there is no required for the glycogenolysis. Okay, when high ATP molecule, when glucose 6 phosphate molecules are high, there is not required for this conversion. Okay, so allosteric activator, if AMP is present, ATP is low levels and AMP is high levels, then it will activate the activation of enzyme by phosphorylating at serine 14 amino acids. So it will give active form activation of phosphorylase by activation of what it called phosphorylation of this enzyme at serine 14 amino acid. Okay, phosphorylase binds to cofactor pyroxyl 5 phosphate and which requires activity. So pyroxyl 5 phosphate is the cofactor for this enzyme. So that requirement, if pyroxyl 5 phosphate is not removed, then the enzyme will get inactivation. Okay. So the prosthetic group vitamin B6 derivative is covalently linked to the enzyme via Skips base formed between aldehyde group and epsilon amino acid of lysine 680 amino acid. So this enzyme always acts as non-reducing end. 1,4-glycosidic is cleaved by phosphorylases with retention of energy potential in the phosphate ester 1 phosphate. So here you can see how these glucose molecules are removed from each branching points in the presence of glycogen phosphorylase. So all branch points will interact by this enzyme where non-reducing end is there. That glucose molecule will be removed as glucose 6-phosphate, and once branching point very close, at least 4-5 glucose molecules are there, debranching enzyme will cut this one and that will add to this chain, so that these glucose molecules will be removed from the non-reducing end by glycogen phosphorylase enzyme. So here you can see some glucose molecule at the branch point. So these branch point molecules will be just removed from the branch point by simple hydrolysis reaction. So you can see single glucose molecule, single means only glucose molecule here. What I mean here, from glycogen, our glucose molecules will removed as glucose 1 phosphate, isn't it? So here, these branch points glucose will be removed as it is as glucose form by simple hydrolysis by cleaving this alpha 1,6 bond. Okay. So during this glycogen degradation, at least 10% of the glucose will be removed or will be liberated from the glycogen as simple glucose molecule. Those are having branching chains. So this is another one, debranching chain. Second enzyme is debranching chain, debranching enzymes. Okay, that they will remove the branch when it is very near to branch points. When they will remove the glucopolymer short sugar chain from the it is very near to branch points. Say for example 4-5 glucose molecules away from the branch point. So this branch will be removed and add to this another branch, so that they will be removed as glucose 1 phosphate molecule, and branch point enzyme will be removed as simple hydrolysis, just as glucose molecule. So the branch glucose will be removed as it is as glucose. So this will be enter into blood system, so that glucose levels goes up. So another enzyme in glycogenolysis, third enzyme is phosphoglucomutase. Okay, this is simple mutase enzyme that we have seen in previous. Once glucose is converted to glucose 6-phosphate, and it will convert to glucose 1 phosphate, that is reversible enzyme. That's what we discussed. So here the same enzyme will convert to glucose 6-phosphate. So immediately this glucose 1-phosphate by hydrolysis it will convert glucose molecule, or it can enter into glycolysis pathway for the energy purpose. Okay. Phosphate is converted. It may be simple hydrolysis. It can be converted as glucose molecule to rise the glucose levels inside the cell, inside the system, or it can be taken into glycolysis pathway to get for the energy purpose for cell metabolic activities. So that is about glucogenolysis. Any doubt? Hello?
No, sir. Yes, sir. No, sir.
Okay, so now glycogen metabolism, we have seen glycogen synthesis from when glucose levels are high, and glycogenolysis, that is degradation of glycogen when glucose levels are low. So immediately glucose levels will make up inside the system for cellular metabolic activities to avoid the hypoglucemia conditions. Okay, so now we can see the regulation of glycogen metabolism. Here, glycogen reserves are most important available source for the glucose energy for mammal cells. Okay, so if more glycogen residues, particles are there, so whenever glucose molecule is required, immediately instead of taking food source from outside, immediately they will contact glycogen storage tissues to release the glucose molecule for its metabolic activities. Okay, so storage and utilization are dietary control as well as hormonal control. Okay, so glucose diet rich diet, so glycogen synthesis will activate, and if you are not taking food at all in starvation condition, so it will release the glucose molecule, as well as hormonal control also there to make glycogen or to degrade the glycogen. Okay, so first primary hormones, epinephrine, which is adrenaline. Here it is one hormone that activate glycogen synthesis, glycogen metabolism. Another hormone, glucagon. Another hormone, insulin. Okay, so these two, glucagon and insulin, are synthesized from the pancreatic tissue by different cells that we have discussed. Okay, primary targets of this thing in glycogen metabolism is glycogen phosphorylase and glycogen synthesis. The action of hormones are indirect here. Okay, so dinner at 9 p.m. like if you have more glucose, all these sweets, all those things during sleep, amino acids and carbohydrates and blood sugars will go high when you are in resting period. So higher insulin will secrete from the pancreatic alpha beta cells, and then glycogenesis will occur. So these sugars will convert into storage glycogen. So when you are working, and when you are in a hurry, when you are wake up, okay, so adrenal hormone get activated, and then this adrenaline activation, the activation of some phosphorylase enzymes or cyclic AMP cascade, and which activates some proteins and they get removal of glucose molecule from the storage glycogen. So glycogenolysis you can see in this stage. Okay. So next, glucagon, when low glucose levels are present, glucagon will be released from the alpha cells and primarily act on liver cells. Receptors are on surface cells interact with this glucagon and stimulate glucagon breakaging enzymes and stimulate inhibition of glycogenolysis. Okay, glucagon also block the glycolysis and stimulate conversion of glucose from the non-carbohydrates via gluconeogenesis. That's what I have seen. So, epinephrine, when glucose levels are low, act primarily on muscle cells, receptors present on these muscle cells, and it stimulates glycogen breakdown and inhibit glycogenesis. Okay, synthesis of glycogen. Okay, glucagon and epinephrine both stimulate intracellular pathway via increasing level of cyclic AMP. Cyclic AMP we have seen second messenger protein kinases, and so that it phosphorylate and dephosphorylate via that covalent modification. Some enzymes get activate and some enzymes get inactivate. Okay, so insulin, as we've seen, when glucose levels are high, it will be released from the pancreatic B cells, and muscle cells and liver cells have the receptors for this, and it will activate synthesis of glycogen. So glycogenesis pathway will get activated during this in this process when insulin levels are high. Okay, so extracellular signal pathway involves complex sequential phosphorylations and dephosphorylation reactions. So cyclic AMP, as we've seen, when AMP levels are there, when ATP levels, when cyclic AMP is present, it interacts with many proteins. Okay, so here you can see epinephrine and interact with the receptors, and it get activate and get activated G proteins, and G proteins activate cyclic AMP synthesis proteins, and once ATP interacts cyclic AMP synthesis proteins, it synthesizes cyclic AMP. Okay, this which is called as cyclic AMP cascade. So once cyclic AMP present active, it binds a protein kinase protein. Okay, so protein kinase A protein get activated by interacting with cyclic AMP, and once cyclic AMP protein kinase, as we seen in in enzymes, allosteric enzymes. Okay, allosteric enzymes activation. Allosteric enzymes are multi subunit proteins, so they will have different way to get activated. Here protein kinase A is allosteric protein, so it has subunit here tetramer. 4 molecules are there. So once they are present as a tetramer, this protein kinase A is in the form of inactive form. So once cyclic AMP interact with that, so it will interact with R subunits, and it will cleave the C unit. Once C proteins are cleaved from the tetramer, these proteins get activated and they make kinase protein. They make phosphor. They act. What I can say, they actively involved in the phosphorylation of some other proteins. Okay, so activated proteins, activated monoproteins interact with the phosphorylases, some proteins, and they phosphorylate proteins. By that way, those proteins get activated and do their activity. Okay, here C monomers get interact with the phosphorylase kinase. Okay, phosphorylase kinase is the protein which interact in the phosphorylase, glycogen phosphorylase, the protein which degrade the first stage. Okay, so phosphorylase is active protein that also that cleave the non-reducing sugars. Okay, so Subunit C also phosphorylates glycogen synthase, inactivate it. So glycogen synthase is the one that involved in the glycogen synthesis. Okay, it is also inactivate this way, and it activates phosphorylase kinase protein P. So this active form will actively interact with the glycogen and remove the glucose molecule. Okay. So how glycogenolysis inhibited? When needed for glucose is over, glycogenolysis is inhibited. A phosphoprotein phosphatase hydrolyze the protein phosphates to like phosphorylase A active form. It will phosphate molecule and it converts into phosphorylase B. Okay, glycogen phosphorylase B. So which is inactive form. Okay, and phosphorylase kinase active form, and phosphate molecule will remove on this structure, and phosphorylase kinase inactive form will be to become phosphorylase kinase inactive form. Okay, by removal of phosphate molecule on these proteins, they become inactive. So how glycogenosis is a complex process is stimulated by insulin. Insulin indirectly activates phosphoprotein phosphatases. Okay, glycogen synthase D. When it is phosphorylated, it is less active. Okay, glycogen synthase D, when it is dephosphorylated, it become active structure. Okay, so by phosphorylating phosphorylation protein, removal of phosphate proteins, it will become active form. So insulin will dephosphorylate and make it active, so that it will synthesize glycogen. So another way, in active muscle, there may still be high glucose 6-phosphate. Less active phosphorylated glycogen synthesis can be activated by high levels of glucose 6-phosphate. It is bypass hormonal regulation in muscle particularly. How is inhibited? Epinephrine and glucagon inhibit the glycogen synthesis. Two ways. One is protein kinase A subunit C phosphorylate glycogen synthase, so that it will inactivate. Another way, phosphorylase kinase can also phosphorylate glycogen synthase, inactivating it, so that it will get inactivated. So summary of this regulation: epinephrine and glucagon stimulate the glycogen synthesis. Okay, via cyclic AMP. Glycogenesis is stimulated by insulin, a pathway ending to dephosphorylation, and glycogenolysis is inhibited via dephosphorylation takes up glucose. So there are many abnormal conditions if you have abnormality in this glycogen metabolism. Okay, a family of series, although non-necessarily fatal diseases, caused mutations in these enzymes involved in the glycogen breakdown. Okay, so here you can see many enzymes are listed, and which are involved in the glucose metabolism, and abnormality of these particular enzymes, and name of the disease also listed here. So of course they are not fatal, but some kind of problem, some kind of abnormality of this organs can be noticed in this condition. In this type of mutations in these enzymes, you can see enlargement of liver loaded with glycogen, less liver, all those things, liver abnormalities you can see here. Okay, so that's about glycogen synthesis. Any doubt here? Glycogen metabolism and its regulation. Any doubt?
Oh, sir.
I didn't put my video. Okay, okay. Any question?
Sir, I have one question related to our academic. Can I ask? Yes.
Go ahead.
Okay. So actually, sir, our mode of instruction is in English only. So can hello? Am I audible,
Yeah,
sir?
yeah, yeah. You
Actually,
are audible.
our
Repeat that.
actually of instruction is in English only.
Right.
So can we get letter from our department or from the academic section that we have a study in English so that instead of IELTS score we can submit I can submit this letter to foreign university, sir. Actually, most of the foreign university are considering letter also. So can
Yeah,
I get
yeah,
it from the
yeah, yeah.
academic?
You can get it. You can get it. You write to head office, okay, HOD office, okay. They will give you a letter that language of this course in English, something like that.
Okay, so HOD office means to you, sir, or
Yeah, contact. You send contact.
okay,
Okay, they will prepare a letter and get a signature from HOD.
okay, sir.
Okay, that letter is enough for you.
Okay, sir. Okay, thank you, sir.
Hmm.
Okay.
Any other doubt? Okay, then, if you don't have doubt, we'll sign off. You can